All right, so today we are going right back to the beginning. I mean, the absolute fundamental unit of human life, the cell.
Hmm.
And you might think you know the basics, but we're doing a deep dive into Chapter 7, Introduction to Cell Physiology, because honestly, without this, understanding how drugs work, especially something like chemotherapy, it just doesn't make sense.
That's the critical point, really.
Yeah.
Our goal here is to just quickly unpack the cell's parts, its processes, its life cycle,
Hmm.
but only focusing on the things that medications are specifically designed to hit.
And the clinical stakes are. They're just massive. You take chemotherapeutic agents, they work by, fundamentally altering how a cell functions.
Or disrupting its integrity, or just flat out preventing it from reproducing.
Right.
And the big challenge, the reason this all matters so much, is that these drugs aren't perfect. They lack what we call selective toxicity.
Meaning they hit our healthy cells too.
Exactly. And understanding which part of the normal cell they're hitting, is it the membrane, the DNA, the energy system? That's what lets us predict side effects and, plan our nursing interventions.
Okay, so let's unpack that blueprint, maybe starting with the control tower. Every cell has a nucleus, cytoplasm, and then the cell membrane around it.
And the nucleus is the program center. It's where all
Yes.
the DNA is stored, sequenced into genes.
And the genes are the instructions for everything, basically.
For every single protein the cell needs to make, for maintaining balanced homeostasis. The nucleus is what regulates when the cell divides, what it produces. It's the boss.
And inside the nucleus, there's that little mass, the nucleolus. Why do we care about that?
Because that's where the parts for ribosomes are made. And the ribosomes are, they're like the factory workers. They move out into the cytoplasm, and they become the actual sites where proteins get built.
Okay, so from the control tower to the border wall, the cell membrane.
Right? This thin barrier separating what's inside the cell from everything outside. It's key for integrity, but more importantly, it's the gatekeeper.
And this gatekeeper isn't just a simple sac, is it?
Oh, not at all. It's this brilliant piece of engineering, a lipoprotein structure.
Lipoprotein.
Yeah, so think of a double-layered wall, the lipid bilayer made of phospholipids. And these phospholipids, they're bipolar. One end loves water, the other end hates it.
So it's like having tiny guards standing shoulder to shoulder, and they make sure nothing that doesn't belong can just waltz right through.
That's a perfect way to put it.
Yeah.
That bipolar arrangement gives it incredible control over who gets in and who gets out. And you've also got cholesterol in there, which just kind of stabilizes the whole thing, keeps it from getting too fluid.
So here's where it gets really interesting for pharmacology. The receptor sites.
Yes, these are proteins stuck in the membrane, and they act like specific locks for chemical keys. They're communication portals.
Can you give an example?
The classic one is the insulin receptor. When the insulin hormone, the key, locks in, it triggers a whole reaction inside the cell that allows glucose to get in. So many drugs are designed specifically to either block or stimulate these receptor sites.
And beyond just receiving messages, the membrane has, what, identification tags?
Exactly. We call them histocompatibility antigens, or HLAs. They're genetically determined proteins on the surface that basically stamp a cell as self.
So the immune system can tell friend from foe.
Right. It's trained to protect anything with your self stamp on it and destroy anything that looks like a non-self invader.
Which is why organ transplantation is so tricky. The immune system sees the new organ's antigens and says, nope, that's not one of ours.
Precisely. Organ matching is all about matching those HLAs as closely as possible.
And then there are the actual doorways, the channels, or pores.
Right, these little protein tunnels that let very small things like sodium, potassium, calcium, water pass right through.
And that's another direct target for drugs.
A huge one. Think about calcium channel blockers. They are designed specifically to block those calcium channels. Since calcium is needed for muscle contraction, blocking its entry relaxes blood vessels, which lowers blood pressure. It's a really elegant, targeted approach.
Okay, let's shift focus to inside the cell, to the factory floor itself. The cytoplasm. It's where all the metabolism happens and where you find the organelles.
And first up has to be the mitochondria, the power plants.
They make the energy.
They make almost all of it in the form of ATP, and they do this through the Krebs cycle, which is a process that absolutely requires oxygen.
So you can tell how active a cell is by how many mitochondria it has.
You can. A heart muscle cell is packed with them. A dormant cell, not so many. And if the cell's energy demand outpaces its oxygen supply,
You get that burn, lactic acid.
that's the one. Then you have the endoplasmic reticulum, the ER. There's the rough ER, which is covered in ribosomes and makes complex proteins.
And the smooth ER, which is critical for anyone taking medications.
Absolutely critical. The smooth ER is the cell's detox center. It breaks down toxic substances, including many drugs. This is where the liver's first pass effect happens. The more active your smoothie are, the faster you might metabolize a drug.
Okay, moving on to packaging and cleanup, we have the Golgi apparatus.
That's the cell's post office.
Mm.
It processes packages and prepares things like hormones to be shipped out of the cell.
And then the lysosomes, the cleanup crew.
Right, these are little sacks full of powerful digestive enzymes. They're supposed to break down old, worn-out cell parts in a controlled way.
Until it's not controlled. And that's where that clinical analogy comes in. The one rotten apple spoils the barrel.
It's a terrifyingly accurate analogy. When a cell dies from trauma or lack of oxygen, its lysosomes can rupture, and those digestive enzymes just spill out everywhere.
And they don't just stay put.
No, they start digesting the membranes and proteins of all the healthy neighboring cells. It causes this massive, spreading chain reaction of tissue death. The textbook uses the example of a decubitus ulcer, a pressure sore, that's uncontrolled cell death, with lysosomes actively making the wound bigger.
Wow. Okay, that really paints a picture. So that brings us to how cells control all this traffic, trying to maintain homeostasis.
Right, so getting things in is called endocytosis. Like when a white blood cell engulfs a bacterium, that's phagocytosis.
And getting things out is exocytosis, like releasing a hormone.
Exactly. And to manage all this, the cell uses two main types of transport. The first is passive transport. No energy needed.
And the simplest form of that is just diffusion, right? Things moving from a high concentration to a low concentration.
Yep, like oxygen moving from your blood into your muscle cells. But a more specialized type is osmosis.
Water movement. This is a huge one for anyone giving IV fluids.
Absolutely fundamental. With osmosis, water moves across a membrane to equalize the concentration of solutes. It moves from an area of low solute concentration to an area of high solute concentration.
So it's trying to dilute the more concentrated side.
Precisely. And that's why we have to be so careful with IV solutions. An isotonic solution has the same concentration as their blood plasma. Everything's balanced.
But a hypertonic solution is more concentrated.
Right, so it will pull water out of the red blood cells, causing them to shrivel up. And a hypotonic solution is less concentrated, so water rushes into the cells, making them swell up and even burst.
So that's passive transport. What about when a cell needs to move something against the flow?
For that, you need active transport. It
Yeah.
requires energy, ATP, because you're pushing something uphill against its concentration gradient.
And the poster child for this is the sodium-potassium pump.
The absolute classic. This pump uses energy to keep potassium levels high inside the cell and sodium levels low, even though the reverse is true outside the cell.
Why is maintaining that imbalance so important?
Because that imbalance of ions creates an electrical charge across the cell membrane. It's what gives cells like neurons and cardiac cells the properties of excitation and conduction. Without it, your nerves don't fire and your heart doesn't beat.
Incredible. Okay, finally, let's get to the cell's life cycle. Mitosis.
Right, its ability to reproduce. And different cells do this at different rates, but they all follow the same basic cell cycle.
And this is the final crucial piece for understanding so much of pharmacology.
It really is. The cycle starts with G0. That's the resting phase. The cell is just doing its job, not preparing to divide at all.
It's dormant.
Dormant. Then, if it gets a signal, it enters G1, where it starts gathering materials. Then S phase, where it actually copies its DNA. Then G2, a final preparation step. And finally M, mitosis, where it splits into two daughter cells.
So here's the huge clinical takeaway for chemotherapy.
This is it. Chemotherapy drugs are designed to kill cells that are actively dividing, the ones in G1, S, G2, or M, because those processes are complex and easy to disrupt.
What about the cells just sitting there in G0?
The chemo flies right past them. They're dormant, they're not active, so they are often completely untouched by the drug.
So a cancer cell can just be hiding out, waiting for the therapy to be over.
Exactly. And when those G0 cells are later stimulated to become active again, the cancer can come back. It's why chemo regimens are so long and complicated. You're trying to catch those cells as they wake up. It's also why that 5-year cancer-free mark is such a big milestone.
Wow, that was an amazing dive. We've gone from the cell's nucleus and its power plants all the way to its life cycle, and at every step, you can see how a drug might interact with it.
And it really underscores that the cell's structure, the receptor sites, the channels, and its processes, especially that G0 resting phase, are the fundamental targets and challenges that drive pharmacology today.
So here's a final thought. If chemo works by hitting rapidly dividing cells, and we saw how devastating a ruptured lysosome can be, could we ever flip that? What if future innovations could target the stability of those lysosomes themselves?
You mean trigger them on purpose?
Yeah. Could we develop a delivery system that specifically tells the lysosomes inside only the abnormal cells to rupture, to turn that internal cleanup crew into a targeted executioner without all that collateral damage to healthy tissue?
That is a fascinating question.
That question of cellular integrity is what we'll leave you with. Thanks for joining us for this deep dive.